With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello everyone, the question is which of the following test is not used for testing proteins? So let us see each of the options: Molish test, biuret ninhydrin test, or Milan's test. Number 1 is Molish test. Molish test. So basically what happens in Molish test is it is used to detect the presence of carbohydrates based on dehydration of carbohydrates based on dehydration of carbohydrates. All right, by sulfuric acid, carbohydrates by sulfuric acid or hydrochloric acid, by sulfuric acid or hydrochloric acid, and produces an aldehyde, alright, which condenses with two molecules of phenol, resulting in a violet ring. That means this test is not used for the detection of proteins. So that means this is the right option because not used is there. So that means first one is correct. Now moving on to the second one, that is biuret test. So biuret test is basically used in detecting the presence of peptide bond. Biuret test is used to detect peptide bonds. So basically what happens in this test is a copper plus 2 ion forms in presence of that is peptide bond forms a violet colored coordination compound. Coordination complex in alkaline solution. So that means we know that peptide bond is present in that is proteins, or you can say amino acids. So that means this option is wrong because it is used in testing proteins. Now moving on to the next one, that is ninhydrin test. Ninhydrin test is basically, third one is ninhydrin test. So what happens in this test is it is used, or you can say it gives positive test for that is alpha amino acids and proteins that contains a free amino group, acid group contains free amino acid groups. Alright, so that means amino groups are present in amino acids, which protein have amino acids. So that means this option is also incorrect. Now moving on to the last one, that is Milan's test or Milan's test. Now let us have a look at Milan's test. What happens in this test is, fourth one is Milan's test. This test is basically, it is an analytical reagent used to detect the presence of that is soluble protein. So a few drops of this Milan's reagent is added to the solution, which on heating gives a reddish brown PPT in that indicates the presence of tyrosine residue which occur in nearly all proteins. All right. So this is used in detection of proteins. So that means this option is also wrong, because we have to find out the test that is not used for testing protein. So Molish test is only used for the presence of carbohydrates. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.